Participants, welcome to today's introductory class. I am Professor Anshuman Pragji from University of Kalyani, and this is the first time that I'm going to host this session, this AfriGen platform of bioinformatics. And let me first introduce my staffs here who are also here in this meeting. I believe that, yeah, let me see. Yeah, there are the staff members, Dr. Shima Bishas, uh, Ms. Shrikona Roy, Mr. Anirban Nondi, Mr. Jeet Mondol. They are the staff members of this entire proceedings, I should say. So, and this is an introductory class, and today you have your actual class, and the timing and the schedule are there in the Vula platform. So I believe that all of you have access to that. So my first question, or rather my first topic of discussion will be on databases. And my question is, I believe that all of you have some idea about bioinformatics. So if you do not have any idea, or if you are stuck somewhere, feel free to send us emails, or contact us through Vula, whatever you are familiar with or whatever you're comfortable with. And so we'll be able to help you out, try to help you out. And another thing that I would like to mention here is that this center, this BIF Center of University of Kalyani, it is funded externally by Department of Biotechnology, Government of India. And from this center, we conduct regular workshops and training programs, and we will provide those information in the website. So if any one of you are interested in joining the workshops, that means those workshops are physical workshops. That means there is a one-to-one interaction with the participants. So if you are interested, you can look into our website. You can check our websites. We provide all the necessary details over there. So with this brief introduction, let me talk about the databases, the basics of biological databases. So let me start. So the screen is visible to all of you? I believe the screen is visible to all of you. Okay. So now, biological databases. So what is a database, and why we need a database? A database is basically a storehouse of information. I think all of us have a little bit idea about databases. So it is simply a storehouse of information. Now, why biological database and why biological storehouse is so important? That is because in biological field, there are numerous kinds of data. The most important ones are the sequence and the structure data. But we started getting interest in biological data from early 2000. And this slide is taken from AMBL, and you can see with gradual increase in the amount of data, it's kind of a data explosion. So what are the different kinds of data and the databases we have? We have the genome sequence data. We have the nucleic acid sequence data. The nucleic acid sequence data may be both DNA and RNA. And then their products, that means the protein sequence and their corresponding structural data. There are gene expression databases, protein expression and functional annotation databases, and interaction databases. That means all these types of data are already present. So let me see if there is any question in the chat box. Now, this is a typical question that comes in all of the interviews or the most of the interviews. If you say that you have some idea about bioinformatics, so what is the need for biological databases? So it is the first and foremost one is to store the information and communicate among the peers, then the disseminate the biological information. That means, the thing is that it may so happen that all of us are interacting through the network platform, through a particular network. Now, in my lab, we are working with Parkinson's disease, neurodegenerative disorders, and gastric cancer. You may be interested in these two topics, but how will you get to know that what we are working on until and unless we have a common platform? So this biological database is a common platform so that we can easily share our individual data amongst us. And also, another important aspect of bioinformatics and the biological databases is that, see, bioinformatics is a field which is a combination of different subjects, like biology is there, and information means information technology is there. That means there are both biology and computer science. But not only these two, there are involvements of physics, chemistry, mathematics, statistics, and all the science subjects, all the scientific fields. So it is a completely interdisciplinary subject. Now, the thing is that why bioinformatics is becoming so important these days is that you can perform an experiment, a wet lab experiment. I should not say experiment because bioinformatics is also an experimental biological subject. So you can perform a wet lab based experiment. But for that wet lab based experiment, you must have certain background information, that what you want to search or what you want to look for. That background information is provided by bioinformatics. Until and unless you have a knowledge of bioinformatics, it will really be difficult for you to set up your wet lab experiments. Because if you do not know the background information, in that case, what will happen? You have to perform multiple experiments. And all the wet lab based experiments, they are pretty expensive. All of you know, all of you are familiar with. So the bioinformatics, or the science of bioinformatics, helps you to streamline your wet lab experiments. So that is one of the fundamental aspects or fundamental usefulness of bioinformatics. And that is why, and the prerequisite for that platform, that prerequisite for a particular bioinformatics platform is formation of databases. Now, what we can expect? We may get sequence, functional, structural, and different types of data along with the related bibliography. That means the literature results, and well structured and index. That means there has to be some pointer so that you can fetch the data easily. There should be some cross references, like you will get a demonstration of all these in the next class on Thursday, when my staff members will show you the hands-on things. That means how to access the data. And the cross-reference means each database is linked to some other databases. So that is called cross-reference. And they are periodically updated. Why periodically updated? That is because the experiments are going on throughout the world. So until and unless the databases are updated, you won't be able to get the first-hand knowledge of the information or the experiments that are going on. That means the latest information. And also, you need to visualize the data. So these are some of the important databases. We'll cover some of them in the next class. So what are the different types of data that are being stored? The nucleotide sequence and protein sequence, patterns, motifs, 3D structure, expression profiles, metabolic profiles, and the different types of proteomic data. Now, there are two main kinds of databases. One is called the primary or derived databases. That means they directly store the experimental results. That means, say you are working on a sequencing project and you get some result, you can directly store your data in a primary database. And the secondary database means these databases take the data from the primary databases and annotate the data. That means annotation means extraction of certain features from the particular database. For example, say you submitted a protein sequence, amino acid sequence of a protein in a database. Now, that's a primary database. Now, another database which is dependent on the data that are deposited in the primary database, that secondary database, what it does is it extracts the sequence information and analyzes the sequence and comes up with certain features like its possible structures, its post-translational modifications, if any ligand has its propensity to attach to the protein, and so on. So that is the secondary database. And some of the databases are publicly available, like NCBI, like PDB, and some of the databases are not. That means you have to pay for them. Like one of the most important databases for small molecules, that is called CCDC, Cambridge Structural Database, which is not free. But that database is an extremely important one for drug designing purposes. So what are the different types and characteristics of biological data? Types means you can store the sequence data, you can provide the graphs, images, the geometric information, and models, and so on and so forth. And characteristics mean patterns, constraints, and so on and so forth. In the field of biology is the nucleotide databases. And the databases are NCBI, EMBL, and DDBJ. You will today I will give you a brief overview of all these databases. This is entirely a theoretical class. In the next class on Thursday, you will get a hands-on experience or hands-on overview of all these databases. So the standard contents. So what are generally present in a sequence database? They contain the sequence information, the accession number, the references, the taxonomic information, keywords, cross-references, and documentations. So what are all these sequences? Means these are the letters, like the nucleotide sequences or the amino acid sequences, like ATGC and so on. Accession number means this is a pointer to get the sequence information. References means these are the bibliography. That means where the data are published, were published. Taxonomic information, like it is necessary to construct the phylogenetic information. Annotation. I mentioned earlier that only a mere letter like ATGC, this does not signify anything until and unless you can extract certain information which are relevant to your experiment, which are relevant to biology. Like from the sequence, you can extract, I mentioned earlier, the presence of post-translational modifications, the presence of ligand binding sites, and so on. And I mentioned earlier the cross-references. These are the other databases to which this particular database is linked. So this is a graphical representation of a biological data. That means what you can do, you can use the sequence to construct phylogenetic analysis, to construct interaction network, to build the three-dimensional structure, and so on and so forth. All these things that you can do. This is a high dimensional biological data. This is mainly for network analysis. When you construct a biological network using the sequence information. This is the information of biological data. That means these are the structure information. That means how structures are represented. This is a scalar and vector fields of biological data. This is not so important. This is basically important for those who are working in the field of physics or biophysics, I should say. And the typical formats of biological data. These are the important formats, but nowadays this FASTA format is mainly used. FASTA, PIR, and this ASN. These three formats are the most important one, and among them, FASTA format is the best. Why? Because the FASTA format is accepted by all the tools, all the servers. So this ASN is called abstract syntax notation. It is generally used by NCBI. It looks like this. First is the sequence entry. That means it gives you the idea about the sequence entry. Means it is mainly the accession number, then the phylogenetic information if present, then the description. That means what the protein or the nucleotide is, then the publication, then the article. That means where the data were published, and the cross references, and so on. This is another information. This is used in UniProt, EMBL. This is a little bit more user-friendly, because here ID means this is the accession number, and it gives you an idea about what type of data are present. It may be a DNA, which contains 237 base pairs. And this is the accession number. This is the description. This is PubMed. PubMed means it's a literature database where you get the literature information of this particular sequence. This is the product. This FT means feature table. Feature table means these are the annotated parts. It is a ribosomal 18S ribosomal RNA. See, when I told you in the beginning that the databases contain the sequences, but a mere sequence does not give you any idea unless you know what it is. So in this case, you get this sequence. This is the sequence, but what it is actually, it is an 18S ribosomal subunit, ribosomal RNA, and it contains these many base pairs, and these are the number of nucleotides that are present in it. Now, the most widely accepted format is this. This is called FASTA format. This is a very simple format. It starts with this greater than sign, and here the first line where you have this greater than sign, this first line represents the entire description of the sequence. Here, this is the description of the sequence, and this is the actual sequence. This is GCG format. This is not used these days, because this GCG format was previously used to construct the phylogenetic tree by a GCG package, but GCG package nowadays is obsolete, and we can construct the phylogenetic tree by using the FASTA format. And this is called GenBank format. This is quite similar to the NCBI format. It additionally, it contains this locus and this origin. Locus means the gene locus, the chromosome number, and the origin means from where, what is the first base. And here this base count is present, and the end of the sequence information is expressed like this. So if there is any question in the chat box, let me see. Okay, so there are two questions I see. What are wet lab based experiments and obsolete meaning? Okay, so wet lab based experiments means, say, a sequencing experiment, uh, like a Sanger sequencing or a next generation sequencing experiment. And this is and wet lab means you are performing the experiment, you are performing the analysis in a laboratory by physically handling the data. For example, say you have taken a bacteria, say E. coli, and extracted its DNA from E. coli. So that is a wet lab based experiment. And obsolete means which is no longer valid. Is that clear? I think it's from Kanvi. Kanvi, is that clear to you? Okay. Uh, so there are no more questions. So let me continue. Yeah. This is called the PHYLIP format. PHYLIP format means, as I mentioned earlier, that this is nowadays this is not used. But why I'm telling you is just to give you an idea about what are the different types of formats you have. And in the interviews or in the exams, like if you are interested in sitting for any of the competitive exams, these types of questions come: what is PHYLIP format and what is its utility, and so on and so forth. PHYLIP is a tool to construct the phylogenetic tree, and this is a format. This is the format of PHYLIP, which you can use to construct the phylogenetic classification or phylogenetic distribution. But as I mentioned earlier, that nowadays you don't need to study so many file formats. That is because the FASTA format is sufficient for all the bioinformatics applications. Now, if you are interested, you can note down these couple of websites. So the important software tools are present here. You can get the information about the important software tools that are available, and followed by their descriptions, short descriptions. And for the database summary, you can look into this particular paper. Now, um, let us come into this NCBI. NCBI is the comprehensive database. The most important one is GenBank. Um, you need to keep in mind that it is not GenBank, it is GenBank, and it has got many different resources. And again, it's a simple and uses simple web interface. And you can directly submit your data by using these two tools present in GenBank. This is the search engine. You can search using this tool on trades. And these are the associated, that means these are the cross-referenced databases in GenBank. So, yeah, this is an overview of the homepage of NCBI, the PubMed. It's a literature database. It's a part of NCBI. And this is the NCBI sequence database. This is called EMBL. This is also another sequence database. There is no difference between NCBI and EMBL, and there are regular crosstalks. That means they share their information between NCBI and EMBL. And the most important one is NCBI is maintained by NIH, which is in US, and EMBL is an European organization. And this is Japan, DDBJ, DNA Data Bank of Japan. So all these data banks or the databases are identical. That means most of the times you get the same information from all these databases. So the question is why there are so many databases which store the same information. That is because it helps to facilitate the updation of the data. Say a Japanese research lab can easily submit the data to DDBJ. Similarly, a European lab can easily submit the data to EMBL. But there is no problem because all these databases are internally connected. They share their information. And this is UniGene. This is specific for genes. And this is for Saccharomyces cerevisiae, a database specific for Saccharomyces. Now, okay, let me see chat box. No question in the chat box. Now, protein sequence databases. Okay, there is one question. Yes, important question. Yes, in India, there is a database specific, which is IPDS. It is the Indian counterpart of NCBI, and it is funded by DBT. I'm sorry, IBDC. I'm sorry. It is Indian counterpart funded by DBT, but it is also a very good one. And I think all of you heard about the nucleotide genome project in India. So, see, the protein sequence databases, these are the UniProt, PFAM. So it is a universal protein resource. It contains the information from all these databases. And this is one of the examples. This is a specific format, UniProt format. You can type this in the query box. You will get to know all these in the next class on Thursday. And UniProt features, what it has is it helps you to align the sequences and retrieve the sequences. And there is another one that is called PFAM. PFAM means it gives you an idea about the family of the protein, about what domains are present in the protein. So this is kind of a functional database. That means this gives you an idea about the functionality of the protein, what domains are present in the protein. And how you can use PFAM? You can use the sequence search method, and you can use the keyword to search PFAM and get the information. And there is this one, gene indices. It is specifically for genes, and this is if you want to know the entire whole genome information, or the entire genome information. These are gene indices, but again, this is redundant. You can get the same from NCBI. And these are a couple of software tools. This you will learn later. And then the structural database. This is a very vital one. This is PDB, Protein Data Bank. Then there is CAT-SCOP. This is for protein structures. Or rather, I should not say protein structure. This is for protein structure, protein complex structure, protein ligand structure. It contains information about a single protein, about a protein complex, about a protein nucleotide complex, or any other protein ligand complex. You can access using this PDB. And it gives you different information, like the sequence information, domain information, sequence similarity, literature, and so on. CAT. CAT means it is a classification. So from the protein structure, you can classify the protein and to which family it belongs. Like, and what are the types of secondary structures? What are the percentages of secondary structures that are available in the protein? And that gives you an idea about the possible functionality of the protein. See, just by knowing the sequence and knowing the structure of the protein will not help you properly. Uh, yeah. This is the hierarchy. You will get to know all these things. Then scope, and this is this gives you an evolutionary relationship along with the PDB entries, and you will get to know all these details. And this is the PDB. So this is an overview of the databases and that we have, and it's mandatory that you join the Vula platform, and there will be extensive discussion on this databases, and it will start soon. So I think all of you know about it, right? All of you have the Vula platform and have accounts in Vula platform, and so uh, join there and get to know all this information. Will also be there, and so if you if you want to ask any question, you can ask. And the assignments are also important. You will get the assignments in the Vula platform. For the class from my University of Kalyani, the BIF Center of University of Kalyani, will again be on Thursday. The timing is the same. Yes, all the file formats are interconvertible, and I mentioned a website in my slide. You can check that, so that you can convert one format to the other. Yes, the universal tool is there.
Excuse me, sir.
Yes.
So, as you said, this file format is obsolete, not in use. So, what is the requirement to learn about it if it's
Yeah.
not in use?
Yeah. So what is the thing is that Philip is a tool that is still used. So if you are using Philip, you have to give your input in that specific format. But nowadays, there are many other tools available, so that you did not have to convert your data in that particular format. As I mentioned earlier, that the if you have your sequence in the faster format, you can do most of the experiments. But still, some labs prefer Phylip, but there is Mega also to build the phylogenetic analysis. And clustral programs also, they accept the faster formats.
Okay, okay, thank you, sir.
And recorded version of the lecture, yes, I believe it will be uploaded in the Vula. PDB CATSCOP, yes. In the PDB, you will get to know the details of CAT and SCOP. They are there. So in the next class, you will have a hands-on demonstration of NCBI PDB. So let me see. Okay, so I believe that if there are no more questions, then let us conclude the session, and we will timetable for these live classes. Whatever the no. The question answer session uh, is at 4:30 p.m. Indian time, and it will not be by me. It will be by the presenter there. I will also be present. Like if you find any difficulty, I will also be present there. Okay, so let us conclude right now. Hope to see you all, and make sure that you check the study material in Avula. See, Kanvi, all these questions, you can send all these questions to Cindy, because she's handling everything. Okay, so see you all. Thank you very much for joining, and hope you had a good time. Thank you.